I'm on day 3 of my sleep experiment, sleeping on the floor, and I feel like it's actually doing some good. I used to wake up with some stiffness in my muscles and not feeling so good, um, but I feel like it's actually doing something. I was quite skeptical. Initially, I thought I would have more stiffness or sleep in funny positions or roll around and hit things around me. I don't have much space on my floor, um, but I'm just sleeping on a towel on. It's a carpeted floor, just sleeping on a towel and trying to actually sleep just looking straight up. Um, that just seems to be how I'm comfortable, and it seems to be doing some good. So I'm pleased about that. Also getting lots of sunlight. I was wearing a lot less clothes than this earlier today. Even though today it's been quite cloudy, but I still try and expose myself as much as I can. Another good thing is I am soon going to be starting my I'm doing a work placement over the summer, and I will be starting that on the 30th of this month, so quite soon. And I will be looking at metabolic signatures of glioblastoma and a few other things. So it's mainly bioinformatics based. So using all those heat maps and the genetic profile heat maps and all that stuff, and the metabolic profiles, and seeing what that means. Also looking at degree of risk of certain agents, so looking at things like smoking and how that affects the brain. Also other compounds of interest that have been linked to glioblastoma specifically. It's always glioblastoma. Other brain tumours don't really get that much research because they're less common. So there's less to look at, less samples to look at. But yeah, you do look at particular cell lines, but that's a bit different. But yeah, just in general, the other cancers are looked at less. I noticed, just research-wise, it's also quite common to look at neuroblastoma because it's quite common in children. And in women, meningioma is like fairly common. It's rare, but fairly common in general. Amongst women, it's common. So that's interesting. That's an interesting thing. There are some differences between male and female brains, and that will have a difference on prognosis. Women have more progesterone, so that can be neuroprotective, and it can sometimes, not always, it appears to have a prognostic impact whereby women seem to do better in general. So that's interesting. Other things, just looking at IDH1 mutations. I didn't have that, which is supposed to be bad. And IDH1 wild type. It's supposed to be that if you have wild type, you have a worse survival, and if you mutant, not like the X-Men, but mutant it's supposed to be a good way. If you're IDH1 mutant, that means that you'd have a better survival, apparently. Metabolically, it's different as well. That phenotype just means that it takes up certain substrates more than others. So that's the kind of idea of it. It's to do with the Warburg effect, and it's to do with the TCA cycle. So yeah, it's an enzyme in the citric acid cycle, the TCA cycle, and it's to do with how it's functioning. The other thing, which is very important, is fumarate hydratase, which is also in the TCA cycle. If it's inhibited in cancer, that's not a good thing, because it means that, fumarate hydratase is, as it's hydratase, it means it's an enzyme. It's an important tumour suppressor enzyme, has tumour suppressor activity. And how do I explain this? And yeah, so if it's inhibited, if that enzyme is inhibited, as it is in many cancers, that's not a good thing. And we can restore function of that in a number of ways that I learned about on this conference. One of the ways that they didn't speak about, which is new science, is what I'm looking at. Not in the lab, but just it's an interest of mine that other people are looking at. And I'm doing experiments on myself, as I always do as a human guinea pig, is using deuterium-depleted water to restore function of fumarate hydratase, if there is dysfunction there with me. So, yeah, restoring function of fumarate hydratase will allow that tumour suppressor action to do its thing and to restore function of those cells which have become defective, become abnormal, meaning they proliferate and just uncontrolled growth, which is what cancer is. So it's interesting how you can do that by drinking water. Some cancers are more sensitive than others to that. Astrocytomas are actually not as sensitive as other cancers to deuterium-depleted water. However, if you have water that is depleted to such an extent as I have, the 25 parts per million, and if you're doing that continuously, then there's no means to say that won't be effective. It might take longer, but it's worth pursuing and thinking about. The main case study that I saw was similar to that in a way, even though the lineage of the cancer is different, is a paper of a trial on a number of lung cancer patients who had metastatic disease that resulted in brain mets. So that had been a primary cancer of the lungs that had spread to the brain, and through drinking deuterium-depleted water, there was stabilization of disease, and I think there was also a partial response in one patient. It was only a small sample, so it's not something that is significant enough, but it's interesting, very interesting, in fact. So I'll put details of that below somewhere, and we need more human guinea pigs doing this. Hopefully not just me in future. And it would be good to see people with active disease doing this, visible active disease. Because I don't have a baseline to work on looking at scans, because I still no longer have no visible or detectable disease. But I treat my situation in the way that I believe if I stop doing what I'm doing, or if I relax and don't pursue all these prevention strategies and protocols, that the disease could come back. I do see it as a chronic condition that you can manage over the course of a normal lifespan, then something that is inevitably going to bring about my demise, my early demise, so that I'm going to succumb to. I don't want to succumb to it. I would like to do what we used to seem to do, which is die of old age. We don't seem to do that so much anymore in this country. So yeah, I'm very excited about my work placement coming up. Sleeping on the floor has seemed to normalise lots of my negative behaviours throughout the day, which is just, I guess, because I'm not holding a lot of tension in my body. Sometimes if you hold a lot of tension in your body, it seems that it escalates upwards and even influences your thoughts and feelings, and then those thoughts and feelings influence everything else. So yeah, it's interesting how that has impacted me in that way. Maybe it's just that I have a nice routine going, or maybe I'm just more mindful about my sleep. Being on the floor forces you to think about your body. And the other thing is, when you're on the floor, I've noticed just generally, instead of, say, if I'm seated, if I'm on the floor, I'm naturally moving around. So if you're uncomfortable in one position, you'll naturally move to another position. And if I'm sat in a chair, my hamstrings are shortening and I don't feel... And it's your whole posterior chain is impacted by that, because everything moves upwards or downwards wherever you have a problem. So posture is a very important consideration at the moment for me. Another thing, I'm writing about the Cancer and Metabolism conference I attended recently. There's a lot to write about, so hopefully it's not too boring, but I've started to write about that, and it's not boring, it's very interesting, but it's communicating that in a way that is easy to read, and that I can convey those complex concepts that were introduced. That's the challenge. For example, I mentioned neuroblastoma earlier. Neuroblastoma appears to be a real glutamine hog. So inhibiting enzymes involved with that energy kind of pathway. Pathways involved with that in that kind of utilization, that biosynthetic pathway, utilization of that substrate is an important target that we need to look at, not just as a primary fuel for that particular type of tumour, but also because most tumours, as Harry Eagle outlaid after Otto Warburg spoke about glucose, just how important glutamine is as well for proliferation of cancer, most cancers, if not all, as a secondary substrate usually, but also primary in some circumstances over and above glucose. For those cancers, that's what we need. Another thing is, in the last video, I spoke about expression of ketolytic enzymes, and I kind of forgot that I wrote a big kind of critique on a previous study from a couple of years ago, I think it was, or maybe a year ago, on a study saying that tumours could use fatty acids for energy. But in the article that I wrote, that I forgot about, but it makes complete sense and I just didn't think too much about it, is that these cancers were using fatty acids for energy, yes, but they were using pro-inflammatory fats. So that makes complete sense, and I'd just forgotten about that. Sometimes you get carried away with stuff and try to compromise too much with people, with people who have different ideas to you, because you want to not be biased and be balanced. But in this case, I think it's perfectly reasonable to look at how these studies are implemented and then see that, well, actually, maybe that wasn't the best method, and maybe these pro-inflammatory fats that they're using are, that's why the tumour is using these for fuel, because when you use the long-chain omega-3 fatty acid, docosahexaenoic acid that you get from oily fish, and, mostly oily fish and algae, seafood mainly, that it actually inhibits these processes involved with inflammation and proliferation. It doesn't appear to be able to use those fats for energy. So maybe these ketolytic enzymes are being expressed because the tumour is able to take up those pro-inflammatory fats. So that's my thoughts. Those are my thoughts, and I'm really excited about this worker placement. I've worked so hard for it, and I hope to keep you up to date with that. And I should be finished my summary of my thoughts and feelings of the Cancer and Metabolism Conference. And one of the main things I was excited by from that is a collaborative effort from different institutions across Europe, bringing together these different metabolic areas looking at cancer. So looking for biomarkers so that we can look at the tumour microenvironment and see what's going on, but also be able to predict not only future occurrence, but in benign tumours, be able to look at how they progress to being malignant, and the different metabolic signatures of those tumours, and just to get it as early as possible, and to see over time those bioenergetic changes, which is what I have been able to see with my tumour using MR spectroscopy. Every tumour has a different profile with that high signalling activity, those peaks and troughs that you will see on MR spectroscopy. So you could look at a tumour on an MRI scan, this special MRI scan, and you would be able to see what kind of tumour it is just from the scan. By how it's expressed, the different what would you call them? Yeah, the different fuels that it's taking up. um, and they correlate to different things. One difficulty with that is you could get some activity from brain damage, but you could work out what that is over time, and also from just experiences. So if I've had a major hemorrhage, which I had, it's quite clear with the area that I had the hemorrhage that for a while there was some activity there. We couldn't tell if that was a malignant activity or not, but either two things have happened, where either the malignancy or either the malignancy has become just, it's not there anymore, or the brain has healed, there's been neurogenesis, and the damaged material has maybe been taken away, because cells can do that. That debris has been engulfed, because cells do that. And I had that after my hyperbaric oxygen therapy, so maybe that's helped in that way for brain healing, or for possible malignant activity, or maybe both. We don't know. It's very difficult to tell with these things. The scans can be very ambiguous, but over time you can get interesting theories about what's going on. Time is very useful when you are able to compare many scans. And that's all we can go by sometimes, because you can't go back in there and see what's going on. That's the only way you could really tell at this moment in time. Hopefully in future we'll have better ways of knowing and seeing that. So, that is me for today. I'm excited about day 4 of my sleeping on the floor experiment. I didn't think I would be saying that at this point. Thought I might give up at this point. But yeah, looking forward to seeing how it goes. I'm doing this for 30 days and my sleep is going well. Tomorrow morning I'll do some exercise and I'll try and keep that up too, get my body composition in a good way. So, have a good day whatever time you're watching this, and stay healthy.